Hey everyone, welcome back. So once again, I just want to check in and make sure you have been following along with your vocab notebook. This is really, really important because we are going to be talking specifically about COVID-19, which means we're going to be talking in more detail. So this assumes that you understand the basics of viruses. So specifically the lytic and lysogenic cycle for viruses. Um, Also, I just want to make a note. This does look like a slightly different setup because I'm doing this on a different computer. There are some videos in this presentation, and so I want to make sure that the audio works so that we can include the videos in the presentation. So I'm going to be leaving. I'm going to be leaving us not in present mode at this moment, and hopefully, when we do watch those videos, then we can go full screen and actually watch them together. But I think it's important to also see my face on the screen as well. At least that's what I'm told. So let's talk about specifically the COVID-19 virus. So, first off, there are different types of viruses. So, for example, this right here, the bacteriophage, this is a virus that infects bacteria cells. This is a version of the herpes virus. We have a respiratory type of virus over here. This influenza is the common flu. And then, of course, we are talking about the coronavirus, specifically COVID-19. So I wanted to show you that there are different types of viruses that will infect different organisms, different types of cells within the organism. They come in different shapes and different sizes as well. So specifically, we are talking about COVID-19, which is a type of coronavirus. Now, when we are talking about coronaviruses in general, it is known as a retrovirus, this right here. And that is because inside of this particular type of virus, inside of that capsid, it has its genetic information in the form of RNA instead of DNA, which adds an extra step for when that virus tries to infect one of our cells. So when we are talking about this coronavirus, like I said, it is a retrovirus. So what's happening, if you look at this diagram over here, we have the glycoproteins around the outside here. They attach to the cell membrane and the capsid enters into the cell. So inside of this capsid is not only the RNA genetic information for COVID-19, but there are other enzymes and other proteins in that capsid as well, so that we can take the RNA and turn it into DNA. We have to reverse transcribe it. So there is an enzyme, RNA reverse transcriptase, that is inside of that capsid. It is another protein. Enzymes are a type of protein. And basically, it will take the mRNA and reverse transcribe it into DNA. So once we have that DNA, that viral DNA can integrate itself or combine itself into the host DNA and just like hide out there and chill there for as long as it needs to. So it can lay dormant for a couple of days, a couple of hours, or it can lay dormant for years. So, for example, with the HIV virus, it can lay dormant for many, many years. With the HPV herpes virus, especially like cold sores, when you see the outbreak, that's when it's in the lytic cycle. When you don't see an outbreak, it's in the lysogenic cycle, lying dormant. So, of course, once it gets the environmental trigger to start producing more viruses and enter the lytic cycle, then the cell will start producing more viruses to spread the virus. So, to help you better visualize what this process looks like, we are going to be looking at this video of the HIV virus. So I know it's not exactly the COVID virus. However, this video does give a very good animation of how this works. And HIV is a retrovirus, just like COVID. And so they do work under the same procedure.
AIDS is caused by the human immunodeficiency virus, HIV. This is an enveloped virus. The envelope is derived from the host cell membrane with viral glycoproteins such as GP120 and GP41 inserted into the membrane as the virus leaves the cell. The core is composed of P24 capsid protein, and the P17 matrix protein helps maintain viral structure. Inside the core are two identical copies of single-stranded RNA viral genome and three enzymes: reverse transcriptase, RT, protease, PR, and integrase, IN. To establish infection, HIV must first attach to its host cell. Attachment occurs by interaction between GP120 on the surface of the virus and the CD4 antigen receptor on the surface of the host cell. In addition to the CD4 receptor, there must also be a co-receptor on the host cell.
So what you're seeing here is the receptors on the cell, the outside of the cell membrane of the host cell. They are specific to the host. So they are there to interact with other molecules sending messages. And that makes sure that those molecules that are sending the message belong to the host. So the virus is essentially tricking the host cell into thinking that it is a safe molecule to let enter into the cell. Now, this co-receptor over here, this is the one right here, that is going to change cell to cell or organ to organ. So that co-receptor is going to be different in our lung cells versus our liver cells versus our skin cells. And so that co-receptor, the fact that the glycoprotein will bind with the co-receptor is why certain viruses target certain types of cells. So the COVID virus, COVID-19, tends to target lung cells over something like skin cells.
Receptor differs for different host cell types. In T lymphocytes, the co-receptor is called CXCR4, whereas on macrophages, the co-receptor is called CCR5. After attachment, the viral envelope and host cell membrane fuse, resulting in entry of the virus into the cell.
Remember, that's endocytosis.
Once the RNA is released into the cytoplasm of the host cell, reverse transcriptase makes a DNA copy of the viral RNA genome. As the DNA is being formed, reverse transcriptase degrades the RNA strand. A complementary DNA strand is then added by the reverse transcriptase, and the ends of the resulting double-stranded DNA segment are joined non-covalently. Treatment with nucleoside analogs or reverse transcriptase inhibitors interferes with these steps. The resulting circular DNA is then moved to the nucleus and inserted into the host cell chromosome by the viral integrase, IN, enzyme. The integrated viral DNA is now referred to as proviral DNA. Following integration, the proviral DNA may remain dormant, or with host cell activation, RNA may be synthesized from the DNA, yielding messenger RNA and viral genome RNA. Viral messenger RNA is translated, yielding viral enzymes and structural proteins. Some of the functional proteins are formed by cleavage of a long polyprotein by the enzyme protease. Protease inhibitors interfere with this step. GP41 and GP120 are inserted into the host cell membrane, and the structural proteins surround the viral RNA to form the core. Finally, the virion is released by budding.
All right, so hopefully that gave you a good visual of how this process works in terms of how a retrovirus can infect a body cell. Oh, no, we don't want to play it.
AIDS is caused by the human
Here we go. So this diagram right here, if I can get my face out of the way. This diagram right here is like an overview of how the retrovirus works. So we start with the spike proteins that we talked about, the glycoproteins binding to the surface of the human cell, so to the cell membrane. And from there, the viral genome of RNA gets released into the host cell. So once it is inside the host cell, it is going to use proteins to basically replace it into DNA. From there, we can make new copies of this viral genome as well as the proteins that it needs inside of the virus itself or inside of that protein capsid. And then as the viruses exit the cell, the dead cells that are left over from the virus using it to reproduce, for COVID-19, because it's infecting lung cells, those dead cells are going to build up in the lungs, and that's why it's hard to breathe. So as the virus kills more and more cells in the process of producing more viruses, those dead cells basically build up in the lungs, and that's part of the reason why it's hard to breathe, as one of the symptoms of COVID-19. Now, just as another example of this, when you get the common cold or the flu, strep throat, that sore throat feeling you get when you're sick, that's because that virus tends to be infecting the cells that line your throat. And so as those cells die, it basically leaves raw tissue to interact with the environment. And so that's why it starts to hurt, because it's raw tissue. There's no like mucus or anything coating it. There's no layer of dead skin or dead throat cells coating it. And so again, that's why you'd feel a sore throat. In the case of COVID-19, it's hard to breathe because it's taking up space in your lungs. All right, so this video here is meant to go into more detail about how COVID-19 will infect a human cell. So this is taking what we saw from the HIV video and just making it a little bit more specific and applicable to COVID-19.
Researchers from China have used cryo-electron microscopy to show how SARS-CoV-2 infects humans. The study published in Science says the virus targets a type of receptor found on human cells in the lungs, heart, kidneys, and intestines. Citing Kingston University microbiologist Mark Fielder, Sky News reports that the virus seems to attack two types of lung cells: goblet cells that coat the respiratory tract with mucus, and ciliated cells that usually filter out pathogens. A previous study published in Science found the virus spike protein has two receptor binding domains, or RBDs, facing downward and another facing upward. These allow the virus to bind with and invade human cells. The virus targets a human ACE2 receptor that has bonded with an amino acid transporter. This subtype of ACE2 structure has never been discovered before. The virus uses the spike protein's up RBD to penetrate the cell. The virus then dissolves its own protein shell and releases its RNA payload inside the cell, according to British Society for Immunology. A study in Frontiers in Microbiology says a coronavirus hijacks the cell's structure to reproduce. The viral RNA takes over the host cell's endoplasmic reticulum to replicate itself and to manufacture the protein parts to make new viruses. According to the Society for Immunology, the hijacked cell's Golgi bodies then package viral RNA and proteins in a viral protein shell. This leads to the creation of new viruses that leave the infected cell via the membrane. The study in Frontiers in Microbiology says coronavirus takeover imposes stress on the host cell. Cell death or apoptosis is the result when the infection overwhelms the host cell's ability to maintain homeostasis.
All right, so hopefully, that's loud. Hopefully that again gave you some good visuals and explanation as to how the COVID-19 virus actually goes to infect a lung cell in the human. So before we keep going, we also have to take into account that the coronavirus overall isn't a new virus to humans. We have been dealing with the coronavirus for many, many years, and I just wanted to include some examples of this. So the common cold that you get, it is actually caused by a coronavirus. It's this name right here, the HCV, so HCoV 229E. That's your common cold virus. It's another type of coronavirus. Back in 2002, there was a SARS outbreak, which is another type of virus, again caused by the coronavirus. In 2004, there was another version of the coronavirus that you see in the common cold over here in Holland. So if you see the HCoV, they are related to each other. Again, you see this other strain of the HCoV 2005 in Hong Kong. There's also a virus called MERS. And again, this was due to a coronavirus. This was in 2012 in Saudi Arabia. And then finally, we get to 2019, Wuhan, China, the immersion of the COVID-19 virus. And so the idea here is that these are all the same type of virus. There's just small mutations between all of these different viruses. So the question is, what are the possible implications of viral DNA mutating? So I'll leave you to think of that on your own. But the one that I want to point out to you, or before we do that, let's do an overview of mutations and what mutations can possibly do. So the idea here is that the DNA, or in this case, the RNA that codes for the genetic information, when we change the DNA or when we change the RNA, that is going to change how the gene is expressed. So if you remember from our previous unit, gene expression and protein synthesis, we express our traits through proteins that are made based off of the code of our DNA. So all of my physical characteristics and features, my phenotype, is all due to proteins being made based off of the code of DNA. So when we do this, we start with our DNA, or in this case for COVID, we start with RNA, we work to reverse transcriptase back to DNA, and then we're starting at DNA. So this would be our genotype. This is the code of A's, T's, C's, and G's. From there, we transcribe it into mRNA. From there, it gets translated into a protein, which is the phenotype. So keep in mind, if the genotype changes, so if that DNA code changes, and again, this could be the difference in alleles, so like blue eyes versus brown eyes, free or attached earlobes, wet or dry earwax, the ability to roll your tongue or not roll your tongue. These are all different phenotypes that have different genotypes, okay? Different alleles. However, like we talked about with mutations, if we change that DNA code, it is going to change the protein, which is going to change the phenotype. So if we are talking about mutating the genetic information in a virus, in this case, the coronavirus, we are going to get a different phenotype of that coronavirus, which is the difference between the other coronaviruses and COVID-19. So what has happened is that because, or what I should say is, if you change the DNA, you change the protein. So that means a protein in the COVID-19 is different than what our body has ever seen. So remember that every protein is going to have a unique and specific job within the body. And if that protein changes, the job or how the protein does its job also changes. So with COVID-19, it's going to code for a specific glycoprotein that's on that coat on the outside of the virus. And this glycoprotein is completely new to our immune system. It happens to match with the proteins on our lung cells, so that's why it's infecting the lung cells, and that's why a lot of the symptoms are like respiratory issues. So because we have this new protein, this new glycoprotein, our immune system hasn't yet figured out how to fight it off effectively. So just keep in mind that this mutation was completely random. It happened, and it happened to infect a person and easily spread to other people. So while this was random, it is dangerous because our immune system doesn't yet know how to fight it off. And so also thinking about this in terms of glycoproteins and thinking about how the virus actually infects the host cell. So if we can think about that process as well as the new glycoprotein that we've never seen before, That's when we can start thinking about how our immune system can react to this virus and how vaccines can help our immune system react to this virus so that we can better fight it off. So I'm going to pause here again, check in with your vocab notebook. And so, what this means is when we come back, now that we have an understanding of the COVID-19 virus, we can now start to look at how our immune system works and how vaccines work so that we can better fight off COVID-19. So, like I said, check in with your vocab notebook, and I will see you back to talk about the immune system.